Please welcome Ranit Aharonov.
Thank you, Yair, for the invitation and the opportunity to speak here. So I joined Pangea 6 months ago. I essentially dropped everything I was doing, and I hope I will be able to share my excitement about the vision of Pangea and the fresh, new, innovative way that it's tackling precision oncology. So let me start with a true story to motivate the excitement. This is a young woman, 35-year-old woman, diagnosed with a very rare cancer. Her disease progressed quickly. She failed several lines of treatment. Her tumor was sent to one of the major NGS labs for testing, and as often is the case, the report returned essentially empty. So no actionable mutations found, biomarkers for immunotherapy were negative, and she continued to progress into stage 4. She reached Professor Raanan Berger from Sheba, who is on our SAB, and he referred her to analysis by Pangea's ENLIGHT platform for treatment matching. And our analysis of her RNA sequencing suggested that she's highly likely to respond both to PD-1, PD-L1 inhibitors and CTLA-4 inhibitors, and this was contrary to all standard biomarkers, and nevertheless, she started taking ipi/nivo and very quickly responded and went into complete remission. 2 years have passed since then, and she's leading a normal life and completely free of the disease. And our hope really is to make this more of the norm than the exception. So we all know that the idea of precision oncology, of targeting specific genomic alterations with new treatments, has actually yielded very, very effective treatments. But unfortunately, the reality is that the dogma of matching those treatments to patients using those genetic alterations or actionable mutations or fusions has yielded very disappointing results. We know that less than 15% of patients have actionable mutations, and even when you do have an actionable mutation, the chances to respond are far less than 50%. So it's not telling us the whole story. Just to give a feel of the numbers, recent clinical studies, including the NCI match, have looked at 13,000 non-small cell lung cancer patients that were screened for close to 20 of these known actionable mutations. Less than 1,000, I think it's 875 patients, actually had a mutation, and the response rate among those was 7.5%, which is essentially the same as standard of care second-line chemotherapy. So as Shai said, there is a huge gap to be met. And the real problem is that we're sort of looking under the lantern, so to speak, because today the dogma is to search for these actionable mutations in single genes, and these are rare. But what is much more common in cancer is aberrations of messenger RNA expression. And what we're doing as Pangea is looking at RNA expression and also opening the aperture. So we're not just looking at the RNA expression of the targets of drugs. We're looking at network of genes that have functional interactions with the drug target or targets, and by understanding the context in which the drug is operating, we can predict whether the drug will be effective, whether the context of the expression in the cells is going to be helpful to the drug or actually more inclined to develop resistance. And the benefit of that, as we show, is that first, we have actionable insights for the vast majority of patients. Essentially, for all patients, we can make a treatment recommendation among targeted and immunotherapies. And our response rates are much higher when patients receive ENLIGHT-matched treatments. So this idea of genetic interactions and looking at RNA expression and these network of genes is based on work more than a decade long, highly validated, highly published in various journals of our co-founders of Pangea, mainly Eitan Rupin, who today heads the Cancer Data Science Lab at the NCI. And I'll try in 2 minutes to explain how this works. So I'll try first to contrast it with the regular or standard way of looking for biomarkers for treatment. So normally what people do is they take data, as Shai mentioned, of people getting a certain drug. Some of them respond, some of them don't respond, and then choose your favorite supervised learning method, look for what differentiates responders from non-responders, and you have a predictor or a biomarker for response. The problem with this is that you have to be very specific in your development, specific indication, specific treatment, and data is still scarce. Data with outcome is very scarce. But what is highly available is big data from cancer without treatment data and follow-up for outcome. And what we're doing is using that data. So there are a lot of in vitro knockout, knockdown, CRISPR, cell line experiments. There's a lot of clinical data of expression profiles of human tumors and overall survival. Again, no treatment data there, and phylogenetic data. And using all of that, we're creating maps that understand, so to speak, the interaction between pairs of genes. So as an example, think of gene A and gene B. An interesting type of interaction for understanding and predicting treatment efficacy is if you know that if gene A is underactive for any reason and gene B is overactive, beyond normal, then gene B rescues the function of the cell. So it reverses the effect of inhibition of gene A. So that's a kind of interaction that's interesting. It's called synthetic rescue, and we create those vulnerability maps. Now, when you have a specific tumor, an individual tumor expression, you can look at any drug, look at the target of the drug, focus in on the network, look at the surrounding genes, and see whether the activation patterns, which genes are up, which genes are down, are such that the drug is likely to be effective or not. And by that, we have an ENLIGHT matching score between every tumor and every drug. And notice that because I do not need any data about humans receiving that drug, I can even make predictions on drugs that were never given to any human just by knowing what they're targeting. So it's enough to know the target in order to make an ENLIGHT matching score for that. So we validated that by looking at 21 blinded cohorts. We set a single threshold over which we say if the ENLIGHT matching score is above that threshold, we're going to say that the patient will respond to that treatment. If below, they're not going to respond. And we took 21 cohorts we never used at all for developing the platform. And what we find is that indeed, if you look at the odds to respond if a patient received the treatment that is ENLIGHT matched, that is recommended by ENLIGHT, versus the odds to respond if they received a different treatment, the odds ratio is 2.59. This is across many different indications and many different treatments. And also we find that even in the background of chemotherapy, our predictions are very strong. So ENLIGHT is a pan-cancer, pan-treatment response predictor. Very remarkably, we also looked at cases where people did develop those very specific supervised biomarkers for a specific drug and indication, and actually our unsupervised pan-cancer approach does as well as those, which was indeed very remarkable. So this is one scenario, but another interesting scenario, so Yair put us there on the treatment decision, but actually we have a role also thinking about clinical development. And why is that? Because if you think of the patients receiving low matching and light matching score, these are also very interesting for pharma. And indeed, we do work with pharma companies because even if you don't have any clinical data yet, we can still create a biomarker for a new drug and predict who is likely not to respond. And these could be excluded from clinical trials a priori. And our validations show that if you compare this strategy of excluding by a low and light matching score to the optimal strategy of having an oracle on your table telling you the future who's not going to respond, we get to about 90 and sometimes more than 90% of that optimal strategy in terms of increasing the response rate of the clinical trial. We're now very focused on prospective clinical trials, so we have close collaboration with the NCI, and the NCI is about to embark late this summer on the biggest of its kind precision oncology clinical trial. And the ENLIGHT platform has been selected as the sole computational platform in this clinical trial. So we are expecting to start collecting about 1,000 patients a year with 5 and counting branches, clinical branches at the NCI. This is also a great opportunity for us to co-develop new technologies and projects surrounding that clinical trial. That case I showed you at the beginning comes from our compassionate pro bono activity. We've done more than 50 patients so far. This was sporadic, and sporadic data is very encouraging. This is not the only case, the one I shared with you. But we are now moving into an orderly clinical trial in order to be able to follow up this activity, and also embarking on clinical trials with many hospitals in Israel. So, as I told you, ENLIGHT is a powerful platform. What are we doing with this? We have 4 products, 2 current and 2 in the pipeline. So ENLIGHT Lens is our research capabilities. We have, as I said, collaborations with 2 biopharma companies and 1 diagnostic company, helping them design clinical trials, look for optimal combinations, enlarging their target population. And ENLIGHT Match is our treatment matching platform that we used as a compassionate one with that patient. What we now found, again very strikingly to us and even surprising, is that we can use deep learning AI in order to skip the need to actually get RNA expression data. So if you take an H&E slide, a standard pathological slide, and you train a deep learning network to infer the RNA expression data, the predicted expression data is rather noisy. But what we found is that if we take that noisy data and use ENLIGHT in order to predict the ENLIGHT matching score, the fact that ENLIGHT looks at networks actually does denoising of the data, and we have very strong predictions also when using that kind of input. So we can directly get an ENLIGHT matching score from a pathology slide, and this obviously opens up a host of opportunity, and we call that ENLIGHT Match DP for digital pathology. And our long-term vision is to move to working on liquid biopsies. This would make a huge shift, so not just treatment matching based on biopsy, but actually monitoring and continuous understanding of whether the cancer is developing resistance, because we could look at the activity of the genes in the network and infer what's happening with the cancer. So let me sum up by just giving you an idea of what's happening in Pangea and how rapidly we are growing. So in March 19, operations started. We signed the collaboration agreement with the NCI and starting developing the ENLIGHT, moving from the academic platform to the ENLIGHT platform. In 21, we had a cell publication, had collaborations, R&D collaborations, and then really since March, or match as it said there, March 2022, a lot is happening. 2 more pharma collaborations, papers. We're about to submit 2 papers, one on match DP, one on a case report. We have a US subsidiary. Many more things are going on, and we just launched Series A. So I think my time is up, and just want to thank the great team at Pangea. This is from, I think, about 3 months ago. We already have 5 more team members and growing. So looking forward for an exciting future. Thank you.